The Snake River is home to one of the Northwest's most important species, the steelhead.
Steelhead are considered a trout. They're a different species than salmon. You know, they are born in the freshwater, rear in the freshwater for a different amount of time depending on the species, go to the ocean, grow up, and come back.
But like most keystone species, their survival is under threat.
They were really struggling for a long time. They're a sensitive species.
Well, steelhead are listed as threatened.
When you have a species that's so variable in their life history, it's so hard to kind of understand, what they're doing and why they do what they do.
It's incredibly important to understand where they are, which helps agencies conserve those locations because we know where the animals actually are.
So how do you find this fish in all of this? Here is how.
We have water.
So eDNA is environmental DNA that we find in the environment. So usually it's when we take a sample of water or soil, and then we examine the DNA that is contained in that sample.
So especially talking about fish, it can take the form of anything. Urine, feces excretion, scales sloughing off, mucus sloughing off. There's really so many sources of DNA.
People have figured out that you can simply pull that water through a filter. That filter captures those DNA particles.
And then extract that in the lab to preserve just the DNA.
The way eDNA works, we are only able to get rainbow trout.
Rainbow and steelhead are the same species. Their DNA is the same. So the only thing that really differentiates them is their behavior. If you're a steelhead and you never leave, then people call you a resident rainbow trout. That's what we're managing. Whether they go to the ocean or not, we care about them.
With ocean conditions, there's not a lot of fish coming back for steelhead. The numbers are low, which is unfortunate. So when they get back here and spawn, hopefully that's successful. And the little ones, when they come out, we need a lot of habitat so that in low run years we get better survival. Within the Palouse Conservation District boundaries, we have a couple of small streams that have potential steelhead use or have documented steelhead use at a very small level.
And we sampled at those because they are all directly linked to the Snake River, and they're streams that in general fish should be able to get up to, and they're streams that we want to do some work in hopefully at some point, and we wanted to be able to say whether or not there were fish in them.
Fish sampling is relatively expensive. Usually you have to send 2 or 3 people out with electroshockers and nets and sampling gear. It just costs money just to drive out there, let alone to then get in the stream and do the sampling. And the more rare the fish is, then the harder it is to prove that it is or isn't there. So when there's 1,000 fish per mile in a river, you have a good chance of catching some of them. And so without too much effort, you can go, yes, this fish lives here. When there's only 2 fish per mile, the chances of you catching those is much, much lower.
I have spent many, many hours sampling in the field in challenging conditions and thinking, I know the species is here, but it's just super hard to find. So I was really excited when I first heard about eDNA, which was in 2008. The first group that really found that we could detect vertebrates reliably in water samples was from France, from Pierre Taberlet's lab. As soon as I heard about that, I knew that that was going to revolutionize the way that we did our work. I started a collaboration with David Pilliot at USGS and Robert Arkle at USGS, and we did our first paper detecting amphibians and streams. And since then, it's just been exponential growth of this field. And so now there's hundreds of papers a year. We've got a journal that's called Environmental DNA. So we have the power now to detect so many more species. I've mostly focused on water sampling to detect rare amphibians. And so that's both endangered amphibians and also invasive amphibians, so trying to detect them early. We've found new populations of different endangered amphibians across the western US.
I've been reading some really cool studies where groups are monitoring ungulates. So they're taking bark from trees and trying to see if there's like deer or elk DNA in the bark, so extracting DNA from bark. Right now, just talking to you, we could extract our DNA from the air around us.
One that I'm familiar with is grizzly bears. They put scent traps, so which is basically something that just smells good to a bear, and then they put barbed wire around it, and the bear just walks under the barbed wire and it pulls the hair out, and then you can sample that, and you can then start to do population estimates because you can look at the genetic makeup of individual bears and figure out who's related to who, and you can get some idea of the population size from eDNA as well.
And we can also be detecting things like pathogens at the same time as we're detecting our target vertebrate species. So looking at that all together is incredibly useful. So it really fills a big information gap that we had. I mean, it's just amazing what we've been able to do. We mainly use what we call a targeted approach to eDNA analysis, which is that we develop assays that will only pick out the species that we're looking for. So we are often targeting like an endangered species. We develop an assay that will just test positive, give us that positive signal only if the DNA of that species is there.
So that's pretty traditional. So once you have those species-specific primers, then you can go out and you'll usually run that water through a vacuum, through a filter, and then take that filter into a lab and then extract the DNA from that filter.
So this is our sample.
While it's difficult to concisely describe the laboratory process, I think that Karen Goldberg gives you a pretty good idea when she says that
it's complicated.
Regardless, once the DNA is extracted from the filter, our results look something like this.
So our eDNA sampling of the 8 sites that we sampled in the fall, 6 of those 8 sites came back as positive for rainbow trout DNA, and both of the sites that came back as negative were points that were higher up in watersheds, so there might be a fish passage barrier in there somewhere. So that could be good means to help us get some more funding to get rid of that barrier. What this will allow us to do is, in the future, when we are wanting to put together grants to do some of these projects, being able to say either, yes, there are fish in this reach, or no, there are not fish in this reach, but we think it's because of this reason, and that's why you should fund this project.
And there's got to be a win-win for everyone, right? We've got to win for the fish, we've got to win for the public, we've got to win for the landowners. And that's where the balance comes in. It's either we have steelhead in this stream and no farming or ranching. We have to have both. But as you educate both the public and landowners on the benefit of steelhead and how it impacts the whole ecosystem, I think that the eDNA sampling is an easy way to show whether there's absence or presence of salmonids, and then really start the discussion.
The most important thing in salmon recovery is what fish are where. That information drives recovery planning.
And so I've been kind of telling people sometimes that the best way to understand how to manage salmon is understanding where are they right now, where have they been, and where are they going. And eDNA gives us a pretty good idea, when used properly, of those different information.
Now, what we choose to do with that information is another level, right? In itself, eDNA just produces information, more information, better information than we had before. But that's all it does by itself, right? The rest of it is up to what we decide to do with those data. I think it's such an exciting field. I think it's like to be able to really understand what's in our water bodies so that we can more effectively target conservation, I think is really helpful and cool. Plus, I just think eDNA is really cool.
It just shows you like how far we've come in the science community, and just the capability we have to restore species and that kind of thing.
We have just begun to scratch the surface of what eDNA can achieve. For the Palouse Conservation District, proving that steelhead live in nearby Snake River tributaries is just the beginning of an exciting new chapter in their conservation mission. eDNA sampling gives us a groundbreaking view into what's happening in our local environments today to help us shape what will soon become the future.